Hi, welcome to Codex. Today we'll be having Matt Herman speak. Matt Herman is the Chief Research Scientist at Fourier Genetics in Austin, Texas. His bachelor and master's degrees are in engineering physics and electrical engineering, respectively, both from SUNY Buffalo. He received a PhD in applied mathematics in 2009 from UC Davis, focusing on applications of compressed sensing under Thomas Stromer. His work on radar with his advisor was recognized with the 2013 Best Paper Award from the IEEE Signal Processing Society. From 2009 to 2011, he was a postdoctoral fellow at UCLA, working with Stan Osher on LiDAR applications. As a senior algorithm engineer at NVU Technology Corporation, he helped lead R&D on the quote single pixel camera from 2011 to 2018. In his spare time, Matt plays the drums in different bands in Austin. And today he'll be speaking on Fourier analysis on the genome. Take it away, Matt.
Okay, well, thank you so much for the opportunity to talk today. This is joint work with Steve Doro, who's on the call with us. And thanks to the organizers for inviting to give the talk. I've watched some of these talks and they're really great. And we want to give our take on Fourier analysis on the genome. So to motivate this, let's take a look at this little video clip here. This is Entacmia quadricolor. It's a kind of sea plant. And you can see it's fluorescing in blues and reds. And you can actually quantify that trait. You can treat blue and red as different features that you can measure, and you can measure the intensity as a function of the genome of that organism. So, and this is basically, that's the idea of what we're going to be talking about. So let's say we have some quantitative trait, T, some organism. It could be a human, it could be a fruit fly, it could be a mouse, or even a crop. Anything that has a genetic code. And let's say that there are N genes that we know that influence the trait. It can also be other factors like the environment, or maybe even comorbidity. A factor that's in a binary state that can be a 0 or a 1. So we're assuming each of these genes and factors only has 2 states. So if it's a gene, there are 2 alleles, or if it's a factor, there are 2 states. And so that means that there are 2 to the n possible genomes that the trait is defined on. So the trait is of size 2 to the n. Each trait has this hidden gene network that's associated with it. The gene network is the network of interactions, how the different clusters of the genes kind of work together and communicate to affect the trait. That has all sorts of rich information, and it turns out that's the Fourier transform that I'm going to define in a moment. But if you think of each gene or each factor as a node, then clearly there are 2 to the n possible different ways in which they can cluster together. So the gene network also is of size 2 to the n. The goal today is really to learn and understand how we can look at different features of the trait by just reading off its Fourier coefficients. So the Fourier domain has a lot of information. And also we're going to be looking at how compressive sensing can discover G, the gene network, even though the gene network is hidden, and even when n gets relatively large. So you can see the size of these objects grows exponentially. So when n gets really large, these things are going to explode. Even when that happens, we can still deal with the scale problem using compressive sensing. So here is an example. On the left, you see a trait. This is a more abstract version of the Fourier transform, but you can see the trait is defined in the gene domain. It's a function of each genotype. And I like to think of the Fourier transform in this ideal situation as an untangling operator. It untangles something that looks quite complicated and dense into a sparse representation that you see here on the right. So now we've entered the cluster domain where the gene network is defined as a function of the different clusters, and it's quite sparse. The representation in both domains is identical. The information is identical, but they look completely different. So this is something that we're going to be looking at. Usually we don't have a full trait, so we don't have all of the trait values. We just have an incomplete data set. So that's where compressive sensing comes in, where we hopefully can take a few samples, random samples, and use that information to recover an approximation to the sparse network. So that's basically what we're going to be looking at. I just want to pause for a second. Emily, I see your face. I don't know whether I'm on the camera or not. So, so that's basically, if we can use compressive sensing to recover a G hat, an approximation, then we have an idea, a representation of the ground truth, the gene network. And that it basically discovers the important needles in the haystack of exponential possibilities. And why do we care about the gene network? Well, that has all sorts of important information of how the different clusters are interacting. That should give us, let us analyze disease susceptibility, designing different kinds of drugs, screening individuals for drug trials, designing crops and livestock that are maybe better, better able to cope in challenging environments, anticipating the effects of gene editing. And also we can take the inverse Fourier transform gene network and pull the data back to the trait domain and be able to predict the average value of traits of the values for unobserved individuals. So this is some of the motivation of why we'd want to do this. So the definition of the Fourier transform of these quantitative traits, it's very simple. It's just a Fourier transform. If you have the full trait, the Fourier transform is this gene network G, and the inverse Fourier transform is on the gene network, and you pull it back to the trait domain. It's defined by these H matrices, which are Sylvester-Hadamard matrices. These are not Walsh-Hadamard. These are actually Sylvester type Hadamard matrices. And the factor, the two to the n here, that's just a normalizing factor, to make this an isometry. We actually put it on the forward Fourier transform side because we want to think of these interactions as averages. So it doesn't really matter which side you put it on. The nice thing is that the Sylvester-Hadamard matrix is recursive and it has a fast FFT-like implementation. Here's a small example. When there's 4 loci, so little n is equal to 4, then we're dealing with a 16 by 16 matrix here. This is H4, and the Hadamard matrices are all plus minus 1s, and these are orthogonal matrices. The first row is actually all 1s, right? So when you multiply this H4 times T, you're going to just accumulate. You're going to get a summation of all the trait values, and when you divide by 2 to the n, we have a regular arithmetic average. So let's look at a simple example to kind of get you on board. We're looking at mouse coat color, and there's 2 homologous pairs. One homologous pair deals with pigmentation. So we're going to say pigmentation is dominant over albinism, and fur color is dominant over, black is dominant over brown. So there are 4 loci, 2 loci in each pair, and that means there are going to be 16 different children. To review, when you have homologous pairs, the left side we're going to say is contributed from the mother, and the right side is contributed from the father, and all these different combinations is what's going to give rise to the different colors of the coat for the mice. So there are 16, you see the 16 children here, and this top quadrant here on the top left are all albino because the contributions from both parents are little p. Both left and right sides of that homologous pair are little p. The other 12 are all have coat color because at least there's one capital P in each of that pair. And of course, the brown mice occur because the fur color contribution, the alleles were recessive, so they're both lowercase f's in these 3 brown mice. Well, let's make this a little more mathematical and convert these to 0s and 1s. So the lowercase alleles are going to be 0s, and the uppercase alleles are going to be 1s. So you can see how we haven't lost any information here. We still have the homologous pairs, and we know what a 0 and what a 1 means in each pair. So we haven't lost any information, but this is a binary representation, a 4-bit code, and we can now convert the 4-bit code to decimal. That's more familiar, possibly. For us, these are just labels, ways of identifying or tagging the different mice. We have 16 mice, and it really doesn't matter whether we go with binary or decimal. And finally, we can now quantify the coat color. We can say the albino is a 0, the brown is a 1, and the black mice are 2s. So we just quantified the coat color. And now we have all the information to put this into a vector or a table, a lookup table. And here we see the trait, the trait for the ith individual is a function of the ith index. And really the ith binary code, which is the genotype. By the way, I always use I for on the trait side. So I is the index for the individuals. And you can see here, the first quadrant here is all zeros. Those are the albino, and these are the other 12 mice, other 3 quadrants. So the trait is defined on the Boolean cube here, and the vertices are the different genotypes. Each vertex is connected to another vertex. That edge there, if you walk along that edge, there's a mutation that occurs. Let's look at this vertex. This first locus here is mutating from a 0 to a 1. So that's a way to think about how the trait is defined on the n-dimensional Boolean lattice. In this case, it's just a 4-dimensional lattice. Well, we have the full trait. Let's take its Fourier transform. So we're just this vector t multiplied by h4, right? We're just doing this first equation here, taking the Fourier transform, and we get g. The factor of 16 that's in the denominator, I'm going to put it on the left side so it doesn't clutter things up here. And so now we've entered the frequency domain. Welcome to the frequency domain. The index is j. J is our frequency index. But it's really a cluster index, and it is identifying 16 different clusters that can happen. And the way to think about this is to look at the binary code. So if you've never seen this before, this is usually what first throws people. And that's why I wanted to show this small example to kind of get everyone on board. The binary codes here, the columns, correspond to the 4 loci, just like the 4 loci on the trait side in the genotype. But a 0 and a 1 mean something completely different over here. When you see a 0, it means it's in the background and ignore it. A 1 means it's like a binary flag that says, hey, look at me. We're doing analysis, right? So that we're basically analyzing. We're saying this is like an indicator function saying this locus is participating in this cluster. So this is locus 1 is in this cluster. This is loci 1 and 2 are in this cluster. This is loci 1, 2, and 3 are in this cluster. So the binary code, it's similar, but it's fundamentally different. And this third column here is just the support set showing which loci are in the cluster. So this is just, it's pretty straightforward. And we have 3 different ways of notating the cluster codes. So we use different ones depending on what we're trying to do mathematically. So I'm going to switch between these 3 different columns at different times. And in fact, let's look at this graph. I'll use the decimal index j because that's a little more convenient to show this graph here. And we see that some of the clusters actually have a larger effect than other ones. And these Fourier coefficients are the weights for the interpolating polynomials, which are the Hadamard waveforms that when you take a linear combination of them and project them back onto the genome, we are resynthesizing the trait. We see the trait as a function of the trait genome index I. Here are the albino, the 0 values, the browns are the 1s, and the blacks are the 2s. So you can see this is kind of, I'm showing you this to kind of get some intuition for how we normally think about signal processing and traditional Fourier analysis. This here is the gene network. It's the same gene network table I showed you a moment ago, but I've permuted the rows. And you can see they're in these 5 groups. And the groups are arranged so that the clusters all have the same size. So these clusters are all size 1, and these clusters 2, size 3, and so forth. And there's a column K, which identifies the cardinality of each cluster. The reason we do that is we call these the levels. So the level K is the collection of clusters that all have cardinality K. So this is level 1, level 2, level 3, level 4, and so on. Of course, if you have more loci, you have more levels, right? This thing grows exponentially. So you have many more levels and much more interesting behaviors when you get to larger problems. But this is a small example just to kind of show you. The reason we put it in this order is because levels are analogous to regular frequency bands that we're a little bit more familiar with. So the low levels are like low frequency bands, and the high levels are like high frequency bands. And here's a Boolean cube. However, this is a different Boolean cube than I showed you before. This Boolean cube is the Boolean cube that the gene network is defined on. So these vertices, the binary codes, are the cluster codes that you see here. And it's very nice because it complements the way the Boolean cube is normally displayed. It complements the level order, right? You can see this is level 0, level 1, level 2, and level 3, and level 4 here. And the number of elements, the number of clusters in each level is n choose k. It's pretty straightforward. So, there are two different Boolean cubes going on, and you have to kind of keep track of which domain you're thinking about. The reason I wanted to show you this as an example is it illustrates the three key ideas that we're going to be talking about today, and that is there's a cutoff level. The cutoff level delineates the low levels from the high levels. So in this case, the cutoff level is basically 2. You can see that they're essentially what we're calling low levels is where the energy is concentrated. The large coefficients are concentrated in level 2 and lower. And the high levels, we expect not to see many, if any at all, large coefficients. So these are the higher levels. Again, with larger examples, you'll see there'll be many more high levels. This is just such a small example. But we have this low level and high level and the concentration into the low levels. And we have this phenomenon of sparsity, or coefficients that are essentially small enough that we can ignore them, and we have compressibility of this vector. So we're going to talk, these are the three main points of today's lecture, and we're going to be coming back to these ideas. Real quick, I wanted to point out we have these weighted simplicial complexes, and it's another way of looking at this. Basically, these are the 4 nodes, the 4 loci, and these are the level 1 coefficients. So I've made them different sizes. The weights correspond to the magnitudes of these values here. And you can see that the fur color homologous pair has a little bit of a less of effect than the pigmentation homologous pair. And then, so that's the level 1 kind of idea, but level 2 are just the edges. So let's look at these level 2. There's an edge connecting loci 1 and 2, and an edge connecting loci 3 and 4, and I've weighted them based on how strong their interaction is. Now there are 4 more edges here that go in between the homologous pairs. So so those edges, if you look, all have value, small value of 1. So they are thinner. They have less intensity, less strength. And if you want, you could threshold that and kind of ignore them. But if they were larger, this is an example of reading off these coefficients in the Fourier domain. If they were larger, that would immediately give us information about how the different loci and the different homologous pairs are interacting. And that might, drive a decision on how you design a drug therapy, or if you're doing CRISPR, because you would know that these genes have, these loci have a strong interaction, and maybe we should know about that before we're cutting things out in CRISPR or something like that. Of course, the higher levels, we then get into these higher dimensional objects. So for level 3, there'll be triangles, level 4 will be tetrahedra, and so on and so forth. So there is a connection with algebraic topology here that I just wanted to mention. So let's get back to the real data and this entacmia quadricolor example that I showed you before. And here is, so we took it from this paper by Polwick, and him and his team, they know a lot about these organisms, and it turns out there's a protein that is responsible for the blue and the red fluorescing. And it turns out that there are 13 amino acids that separate blue from red in this protein. So on the left here, we're looking at a close-up of the protein, and these are the locations on the protein of the 13 different loci. And then, so let's say these are all, and there's 2 states. These are amino acids, and they can be in 2 states. So the states they're in here, we can call these the 0 state, right? These are the arbitrary loci. And so this is all zeros. And when it's that's when it fluoresces blue. And when it fluoresces red, this is all of those amino acids have been swapped out. So this is the all ones genotype. So that's what we see over here. It's arbitrary, the order that we put the loci in, but once we choose an order, we have to keep that fixed, okay? So whatever order they collected the data in, whatever they assigned locus 1 through 13, that corresponds to these different amino acids. And you can flip all of them from 0 to 1, or you can go step by step and take different combinations of these. So it's a real combinatorics problem, right? And that means there are 2 to the 13th different genotypes, so 2 to the 13th different different variants. And that is the data that I got from this paper. So this is the trait. We quantify the trait by measuring its brightness. So there are 8,000 different genotypes that they measured the brightness of. And you can see the majority of the bulk of them are not fluorescing very much. But some of them are, and the ones in this middle range around 0.8 are all reds, and the ones that are all around 1.4 are all blue, okay? So we can use this information. It's great. We have a full trait. We typically do not have a full trait to work with. And so because we have the full trait, we can take its Fourier transform. So here's the full trait, and we can apply the Fourier transform to this. Just multiply it by H13. It's an 8,000 by 8,000 matrix. Divide by the normalizing constant of 8,000, and this is the gene network. These are what I showed you earlier in the talk. You can see it is quite sparse. In fact, if we do a sorted decay, it decays extremely fast. So this is quite compressible. And let's take the gene network and put it in its level order, like I showed you before with the mouse coat color. We now have this in the level order, and this is a more striking example where you see the coefficients are really concentrated into the lower levels here, and there's a cutoff level. I don't know, let's say it's level 5, looks like a reasonable place to say that's delineates the low levels from the high levels, and we see the vast majority of the coefficients are quite small or trivial. Let's look at the top 10 coefficients. These are the 10 heaviest hitters. This is where they occur in the regular decimal order of the gene network. But if we do the level order, we see they are all concentrated in levels 0 to 3, okay? So let's next look at this table just to kind of reemphasize this idea of how to think about the frequency domain. The J index is the location of these 10 strong coefficients in the regular order. And the binary equivalent, the cluster code, wherever you see a 1, that means that locus is participating. So we can see column 4 and column 9 here has a 1, and the zeros mean those other loci are not participating. So only 4 and 9, the amino acids at locus 4 and 9 are participating, and it has a pretty strong interaction effect. It affects the trait in a strong way. Similarly, so these are the top 10. So you can see the top 3 here are pairs of loci. So these are level 2 coefficients. And the next 3 are level 1 coefficients. Those are just single loci, single amino acids affecting directly. And then the next 3 are triples. So amino acids working together as triples. By the way, the index 0, and that's level 0, that's the null set. And we kind of call that the trivial coefficient. I mean, it's not trivial, but it's just the average. So it's 0.538. So let's go back and look at that. So that's this value here, 0.538. That's the average value of the trait. And sure enough, since the bulk of the trait is down around 0.3, 0.538, that looks like a reasonable there. So so everything we've been doing is we've been like reading off features of the trait from the Fourier coefficients, and we stole that phrase from O'Donnell in this text analysis of Boolean functions. And there's more we can do. So we can look at the fitness landscape of a trait. So what you see here is the Boolean lattice on the trait side. So this is the trait, the Boolean lattice that the trait is defined on. So the vertices are all the genotypes, and the fitness here is the height of the landscape is the trait value. So this is something evolutionary biologists are very interested in. And you can think of evolution in some sense as a percolation through the Boolean lattice, or random walks from different vertices to different vertices. And if each random step is a random mutation, then you can kind of see as you're walking around these steps, you're traversing from valleys to peaks and so on and so forth. So, but we can actually quantify the roughness at a locus. So there are n loci, and at locus L, the roughness is the accumulation of all of the differences in the trait when you're looking at these 2 different traits, their difference when they only differ by the symmetric difference on locus L. So the roughness at locus L is all the effects of the differences when there's a mutation locus L. And it's squared because this is a kind of energy, right? Roughness is kind of how the terrain varies. So this is a kind of variance, okay? And then the total roughness of the landscape is just the sum of all the local roughnesses. But guess what? We can do all of this in the Fourier domain. So now I'm going to talk about relative local roughness. And we kind of change the language. We call it influence. And this is just the language that they use in analysis of Boolean functions. It's called influence, energy. These are all different terms that all essentially mean the same thing, but it's the influence on roughness. And the local roughness that I just showed you a moment ago, now, okay, so the denominator is just the sum of the square of all of the different clusters, ignoring the trivial one. So that's just in the denominator for both of these. So you can ignore that. That's just to kind of give a relative, that's just to kind of normalize. In the numerators is what we need to pay attention to. And in the numerator for the local influence or the local roughness, it's the sum of the squares of any coefficient that has a cluster of which L is a participant. So if locus L is in that cluster, as I showed you before, then we take that coefficient squared. And again, the square of the coefficients kind of comes about, you can imagine, because of Parseval's theorem, right? There's the equivalence between the square of the coefficients in the Fourier domain and the square of the coefficients in the original domain, which is the trait. That's kind of why how this comes out. That's the relative local influence on roughness. The relative level influence is a similar formulation, but now we just want to look at any coefficient whose cluster has cardinality k. So all of the coefficients that have all of the clusters that have cardinality k, we look at their Fourier coefficients and square them, and then we multiply them by this factor of k. And the reason we do that is because if a cluster has k loci in it, it has like a k-fold presence. So because of that, we need to, it has an extra weight. So that is what the kth level influence is. And you can kind of see the higher levels have potentially a stronger effect. They kind of can have more influence. So the relative roughness here is just the summation of like we had before, the summation of the local roughnesses, or it's also the summation of the level influences. Key though is this formula here, this expression here. This is kind of like a center of mass. And if you see in the numerator, as I said a moment ago, the higher levels have potentially a much larger effect. To me, that's kind of like an unfair advantage, right? Like if there's any big coefficients in the high levels, they will swamp out the effect or the influence of the lower levels. The converse of that is, what that means is, if we wind up getting something that's smooth, the only way that could have happened is if the higher levels did not have a strong presence. So if it's smooth, it means that the lower levels dominated, which is another way of saying low level concentration. So this RT factor, this overall total roughness, it's going to be in this range of 1 to n. It's easy to show the bounds are between 1 and n. And I put this in a box because we're going to be referring to this quite a bit. So now that I've shown you this, let's move on to some examples. Here is a kind of a simplified view of a dominant trait landscape. And this is to visualize it, to kind of get some intuition. This looks pretty smooth to me. So we have two loci here, right? Loci 1 and locus 1 and locus 2. There's only two loci. So what you see on the base here, this is a two-dimensional Boolean lattice, otherwise known as a square. And as a function of these four clusters, or these four genotypes rather, you can see the trait value. So this is the landscape. The trait value is either a 0 or a 1. And these three corners all have trait value 1. So to me, this looks pretty smooth. Let's see if our Fourier tools bear this out. The Fourier transform, we can see the clusters, the level 1 clusters have magnitude, these are basically, they're all -1/4 of the Fourier. But let's look at the local and level influences. We see that they're all the same. And when we add them up, the total roughness is 4/3. So we're in this range, and little n is 2. There's only two loci. So return to this formula between 1 and 2. where does the roughness fall? 4/3 is closer to one. So I'd say we're fairly smooth. This is fairly smooth. It's a very small example, but what's more interesting is this next example. This is a different landscape for a trait that has this heterozygote advantage. So the only thing that changed here is this fourth genotype. So where this one here is flipping from a 1 to a 0. So this has an advantage for the heterozygote genotypes. The 0, 1, and 1, 0 have a trait value that is 1. So there, that has it's more fit, and it has zeros for the 0, 0 and the 1, 1 case. So you can see 0, 0 and 1, 1 genotypes have trait value of 0, but the other corners have trait value of 1. So this is a heterozygote advantage, and its Fourier transform looks remarkably different. The level 1 Fourier coefficients are 0. And what that means is when we look at the level influence, it has no influence in level 1. All of the influence is concentrated in level 2. And because of that, the total roughness is 2. So now looking back at this, little n is 2, we've achieved the maximum. We've achieved the upper bound. So this is maximally rugged. And you can see visually, this looks much more rugged than this guy. So once we get beyond this of 2 dimensions, it's very hard to show, to visualize these things or to illustrate it. Although I watched Nate Strawn's Codex talk last week, and he had some interesting ideas on how to visualize things. So maybe there is a way to visualize this. But let's return to our luminescence problem that has 13 loci. So we're dealing with 13-dimensional domain, or a Boolean lattice that is, has 2 to the 13th vertices. It's very hard to visualize this. So we can just show a graph of the local and level influence. So the local influence here, we could see 4, 9, and 11 have the largest influence. And that makes sense because going back to even in the top 10, we see that 4, 9, and 11 show up quite a bit. So those are the loci with the most influence. And the relative level influence, we can see level 2 has this disproportionate effect. Again, the top 3 coefficients were level 2 coefficients. So we need to take those, square them, multiply them by the weighting factor of 2 and add them up. I mean, plus all the other ones, but these 3 have the largest effect. In fact, 70 to 80% of this influence here is from those first 3 coefficients. So the relative total roughness, if you add up all of these coefficients, is 2.1. That means we would, if we put our finger right here, 2.1, that's where the balance point, the center of mass on the level influence is. But now there are 13 loci, right? So in the range of 1 to 13, if the total roughness is 2.1, that's very smooth. So this is a very smooth terrain. Okay, this next slide I want to talk about is somewhat speculative. So I want to mention that as a caveat. It's just something we notice an intriguing relationship here that these local influences may exhibit a scale-free network, kind of thing that Barabasi-Albert-Price and many others are interested in. And usually they're talking about graphs and the valency between different nodes. So we want to use roughness as an analog to valency and see if the local influences exhibit some kind of power law relationship. So we're going to look at these local influences, okay, and look at them in their ascending order. So we're going to just sort them in their ascending order, and that's what we see right here. So their ascending order is best fit by this red curve, and but we want to use roughness as our gauge. So we need to flip the axes. We need to kind of invert. That's what you see on the right. Blue and the red are just the inverses of what you saw on the left here. Now roughness is the x-axis. And the red curve is, this is kind of like estimating the density of primes, right? You want to say for a given number, how many primes are there below that number? Well, similarly, we say for a given roughness, how many loci are there below that roughness that have that influence on roughness or less? So for like roughness of 0.3, there are 10 loci that have that influence or that amount of roughness. And then you can see there are these other that have, there are relatively few that have a high influence on roughness. And the derivative of the red curve, this is all empirically, but the derivative of the red curve is just this black line here. And you can see it shows there's a very high density of loci that have small influence and a low density of loci that have high influence. And empirically, we found that equation of the black curve to be x to the negative 0.79. So that is a power law relationship, which is what we needed to show. However, I want to stress this is one example, and it's a very small example. Typically, when they look at these scale-free networks for graphs, they are, the number of nodes is millions or billions and trillions, like the internet. So it's a much more robust kind of network. So this network only had 13 loci that we were looking at. And they also have many, many hundreds and thousands of examples that they average over to kind of take care of outliers. So nonetheless, this is intriguing and interesting to look at this behavior here. The key, though, is just to summarize, we're looking at what we call highly evolvable traits. These are traits that have relatively few loci with a large influence on roughness. That's these loci over here, these large guys. And if we can call them rough loci, then if it's a rough location, then they have they can cause a much larger change on the trait. So because of that, they are kind of stuck. They evolve much more slowly. In contrast, most of the loci have small influence, which means they have a much smaller effect on the trait. So they're freer to explore and adjust to their environment and make small improvements. So that's a way to think of it. But the analogy is like the rough loci are like the hubs, and the smaller loci are the smaller roughness loci are kind of like the spokes. I mean, it's kind of like a hand wavy analog. But overall, these traits have smooth landscapes. And again, the key ideas is that there's a cutoff level, there's low level concentration, and sparsity and compressibility of these gene networks. Just real quick, I wanted to say, what happens if the order of the loci is permuted? So going back, so this is just to kind of think about something a little more mathematical. What happens if these, when they collected the data, it was arbitrary what they called locus 1 and locus 6 and locus 10, whatever it is. This is just an abstract representation of a genotype. It was arbitrary, the order, what they decided, which locus was which. If they'd taken a different order, that would just have been a permutation on the loci. But if you permuted the loci, well, the trait values would have still been the same. So we would have the same outcome of the trait values, but they would have been in different locations, which would have meant that the permutation of the loci would have induced a permutation on the full trait. So it's something to think about. It's when I first showed this to you, you probably saw like these harmonics. You probably are thinking about your traditional Fourier analysis, but I really want to caution you that this is the intuition on Fourier transforms on the Boolean lattice is much less intuitive, because these permutations, we would have had equivalent data, except it would have looked completely different. There are 13 factorial different permutations of the loci, and the data would have looked completely different. It would have not had necessarily this structure. I mean, I've done some experiments, and I did one where it looked like just like complete noise. And you might have thought, oh, it's all noisy, but in fact, there was a lot of structure in there. So you have to kind of suspend your intuition about what that means, about what harmonics mean in this sense, because things that are right next to each other in this kind of decimal flattened point of view is not really indicative of the relationships on the Boolean lattice. And moreover, what does it mean in the Fourier domain? What is the effect in the Fourier domain? If we had a permutation of the loci of the genotype, then that would have meant the same permutation of the loci in the cluster codes. What that means is we would not have disrupted anything about the level representation. The low levels would have stayed the same, except like these three pairs would have been different loci. It would still have been three pairs. So it turns out that permutations that are induced by bit permutations commute with the Fourier operator. And in general, you don't have that relationship. So I thought I'd throw that out there because it's kind of an interesting theory. This is a group on harmonics, Fourier analysis, algebra, and combinatorics. I thought it was a relevant thing to say. So I'm going to pause here for a second because I need to switch who I'm looking at and see if there's any quick questions. And there we go.
So Matt, I did just have one question.
Yes.
So what you've done so far is predicated on having Boolean networks, like when you get to your roughness and so on. I think there's some indication from systems biology that it may be more of a fuzzy logic. Have you looked into the impact of what that would have on your analyses of roughness?
We haven't. It's a really good comment. I will say that we do have some other data from some studies we have on myopia in mice, and in that case, the number of alleles, they're actually each homologous pair has 3 states, not 2 states. So we had to kind of round up to 4 states. So we basically wound up having, we weren't just dealing with binary states is my point. It doesn't really address your question because what we wound up doing is we just doubled the number of loci. So we had a Boolean cube that was, was twice as large. So we're still looking on a Boolean cube and we're not dealing with fuzzy logic. We haven't looked at that. It's something to look at in the future. I'd have to think about what the impact is of that. So
yeah, that 3 level sounds interesting. Is that like a Łukasiewicz logic? You know, yes, no, maybe, or?
No, it's more just that the two, we have an A, a B, a BA, sorry, an AB, an AA, or a BB as what pops up. We never see a BA, and so those are the 3 different states. It's probably just because in the lab, an AB is the same as a BA, so they assumed it was, they just flipped it and said, oh, AB is equal to BA, and they just recorded it as that. So that's why there's 3 different states. It's not a maybe.
Okay, so that would fit well into the Boolean framework,
yes,
just doubling up
exactly,
then.
Exactly.
Yeah.
So that's what, as I started to say it, I realized like, wait, this isn't really a fuzzy logic situation. But anyhow, that's something to look at.
Yeah, thanks.
Let me move on. So next I want to talk about modular traits. And modular traits, there's modular structures all around us in the natural world, in social structures, and in the industrial world, our cars, our HVAC systems. I mean, there's all these subsystems that have different functions assigned to them. And there's a large body of evidence that traits also have this kind of organizational structure. And it makes sense, let's say the genes that are responsible for having good eyesight probably don't need to communicate or interact with the genes that are responsible for having strong lungs. So those might be dedicated to different modules, yet your ability to survive, running away from a tiger, you need both of those to be well developed, yet they don't necessarily, those genes don't need to interact with each other. So to like a good approximation, we can essentially partition a trait into different modules. And this really promotes evolvability because if modules are kind of partitioned in this way, then if there's a mutation or something goes wrong in a different module, it won't have an effect on a different module if there's a limitation in how much they interact. So we're basically going to assume there's no interaction, but of course we can relax that. We can say maybe there's a small amount of interaction across a partition. Maybe if this is like a committee, the committee heads might need to communicate and coordinate, but in general, the number of communications between modules will be relatively few compared to the exponential number of possible interactions within a module. So within a module, however many loci there are 2 to the n. So in this case, n2, there would be 2 to the n2 possible interactions within that module. So let me go to the next slide. And here's a simple example, a simple circuit with 2 modules, and there are 2 loci in each module. So there are 4 loci again. And a lot of metabolic processes are modeled to good approximation with this kind of logic circuit. So you can think of there's some metabolic process, you're in some state and you need to get to some other state, maybe synthesizing an enzyme. You want to achieve some goal. And what we have here are two different branches, two different pathways to achieve that goal. And essentially, in each branch, there are two genes that need to work together. And let's say they both need to be active at the same time in order to achieve the goal. So you achieve success if they are both on or both active or both at the same time. So that's if they're both in state 1 at the same time. So either gene 1 and 2 or gene 3 and 4 need to be on at the same time. And in that case, we achieve the goal. So that's what we see here. This is just a truth table. It should be familiar to most of us. And I've emboldened the trait values where success happens. So the ith trait has success, we call that a 1, whenever locus 1 and locus 2 or locus 3 and locus 4 are true. And again, the ith individual has the ith genotype, just like we had before, and the zeros mean we did not achieve success. But we have a full trait. So if we have a full trait, we can take the Fourier transform, just like we did before, multiply the trait, the vector by h4, divide by 16, although I'm going to put the 16 on the other side to take it out of the denominator, just so it's a little easier to read. And we see, just like we saw before, let's permute this into the level order again. And this is just like the mouse coat problem that we saw before. And this is a modular structure, but it has all the 3 characteristics we like. There's a cutoff level, there's low level concentration, and there are many coefficients that are small that we can essentially ignore. The influences that I talked about before, essentially the total roughness is 1.5, and now the number of loci is 4. So in the range of 1 to 4, we have 1.5 is closer to 1. So we have this relatively smooth landscape as we talked about before. So this small example shows that we have all of the things that we like that we. Just some theory. We have a simple probabilistic model. So let's say there are capital M modules, and so in each module there are n little m loci. So in the mth module, the ratio of loci, we call it alpha m. It's kind of like the ratio is n little m, the ratio of that to the total number of loci. So that's alpha m. So that means the probability of a k loci cluster occurring in that module is just alpha m to the k, basic probability right there. So, but that's just in the mth module. If you want to look over all modules, then we have to sum up all the alpha m to the k's over all m modules. That is the probability of a k loci cluster occurring. And if we want, we can then do some asymptotic analysis on this, and we see essentially if one of the alpha m's is large, it's going to dominate. And that's what this kind of this expression captures here. The a in the denominator is greater than 1, and essentially you can think of that as an effective number of modules. This is an asymptotic result, but we can then, now that we know the probability of a k loci cluster, we can find out the expected number of in level k, because we're talking about k loci clusters. K loci clusters are in level k. So the expected number of them is just the probability times the size of the level. Kth level, how many elements does it have? It has n choose k elements. So this S sub k is the expected number of meaningful clusters. You can also think of that as a sparsity. The cool thing is, this favors smaller clusters. And the reason why is n choose k, well, that rises polynomially. However, P sub k has this exponential term in the denominator. So this decays exponentially. So the exponential decay really just pulls it down. So it favors smaller clusters. The large k just cannot outrun the rate at which the exponential is decaying. So because it favors smaller clusters, that's another way of saying low level concentration. So to see this in action, we decided to go back to the luminescence protein example and look at the histogram of how many strong interactions there were how they were distributed across the levels. So this, we're looking at a bar chart here of the number of interactions that were meaningful in each level. So that's basically this table here, but extend it down to the top 200 instead of the top 10. Look at the top 200, find their index J, look at the binary code, identify which loci are participating, and look at the cardinality of each cluster, and do a histogram on this column K. That is what we are looking at here. This is the histogram of that column K. And we see it's a unimodal and it's concentrated into the lower levels. Level 3 and 4 seem to have the most number of interactions. I will point out though, this is a little misleading because it doesn't show how strong these interactions are. It says that they're the top 200. And in fact, it shows levels 5, 6, and 7 have about 30 or 40 meaningful interactions, but I'll remind you that in the level order, levels 5, 6, and 7 were actually quite small. But nonetheless, about 30 or 40 of them were big enough to be included in the top 200. So, but we said, well, let's apply equation 21, this equation we just showed you here. How does the theoretical expected number of meaningful clusters across the levels, what's that distribution? How does it look? And it actually looks pretty good. We did like a best fit curve here, and we found that parameter A, the effective number of modules, to be 2.4. So out of 13 loci, we found that the effective number of modules is 2.4. I mean, this was surprising because we don't really know if this trait, if this protein actually with the 13 amino acids has a modular structure. It's kind of on the boundary of, is it going to break into distinct modules, or is it more fuzzy, kind of what you were just saying a moment ago? It's kind of hard. In general, it is quite difficult to discern modular structures. And also it's surprising because this is an asymptotic result. So that really should be true. It's more of a faithful result when n is much larger than 13 loci, but still it worked pretty well. So, we don't know if this kind of relationship is kind of pervasive, whether or not there is a modular structure or not. So, okay, so that was looking at the sparsity as a function of level. What about the total sparsity? How does the total sparsity grow asymptotically with n? So as the number of loci grow, how does the sparsity grow with that? So this is the last thing I have two more things to talk about, this and then some compressive sensing. And so this is, we have m modules again, and we want to talk about the cutoff level. The cutoff level is the largest module, the module that has the largest number of loci. So in this case, just to make it easy, it looks like module 2 here is the largest. So N2 is the largest module because it has this largest number of loci. So that's the largest cluster that can interact, the number of loci that can interact in a cluster, because we're assuming there's no interactions across the modules. So N2 is the largest module. And we want to look at 3 cases of what the largest module can be. So if the largest module is a fixed proportion of the number of loci, well, when we do the asymptotics, as we let n grow to infinity, as n grows, the size of the gene network, the total number of interactions, grows with 2 to the n. So that grows exponentially. When we find the sparsity is bounded, it also grows exponentially, but at a smaller rate. The other 2 cases are a lot more interesting. If the largest module, it grows with natural log of n, then we find that the sparsity grows, or it's bounded by a polynomial in n. So that's very interesting. And even better is when the largest module has a fixed upper limit. When it's basically a constant, then the sparsity is bounded by a linear function of n. So the worst case is the first case, and it's probably not very realistic for the largest module to grow unbounded. That's essentially what this says. It's unbounded. It's just always a fixed proportion of how many loci there are. So that's probably not, there's probably a biological constraint on how large a module can grow. But even still, the sparsity ratio, the ratio of S compared to the size of the size of the total gene network, it already is decaying exponentially. So, for reasonably large n, this is going to get vanishingly small. So even though this is not, this is This is this is not ideal. It's still, eventually will decay to something. We'll have a pretty sparse gene network. The other two cases, clearly we have the sparsity is much, much less than 2 to the n, and it's growing polynomially or linearly with n compared to the exponential explosion of 2 to the n. And in some sense, we can think that maybe they are a bit more realistic, but we don't know. This is still speculation. In summary, we have these modular traits that can be broken into these modules and submodules that for tasks and different functionality, these gives us the 3 things that we really want: this cutoff level, this low level concentration property, and sparsity or compressibility. Okay, compressive sensing. Emily, how much time do I have, please?
Maybe like just a minute or two to quickly touch on some things, since
Give me 3 minutes. Well, I assume most of us know about compressive sensing. We have an observation vector y, and we assume that the model, that the observations is the sensing matrix A times the G that we want to discover. So A times G plus E. This matrix A, the rows are just the rows of the Hadamard matrix, HN. And if you remember, so that's just this equation 2, we're making observations and it's just random rows of H. And we have this coherence property in general for a orthogonal matrix. So the number of measurements we have to take is just this mu squared times the sparsity times the log of the ambient space. The ambient space of that we're dealing with is 2 to the n. And luckily for us, for the Hadamard matrix, the coherence is 1. And when we take log of 2 to the n, we get this little n factor. So it's very nice. The number of measurements is just linear in the sparsity and the number of loci. Usually in the literature, the number of measurements they often cite as 4s empirically they found. Most of those experiments are for smaller vectors, not these large ones we have to deal with in our case. However, returning to our data that we only have 8,000 genotypes. So this is actually a very small problem. So we can deal with this 4s as a gauge, as something to rule of thumb. So if we assume we have 200, the sparsity is 200, then if we take 819 measurements, so we've achieved this greater than 4s property, then that subsample ratio of m to capital N is 10%. So this is the subsampling of the original data set. So let's say this is all we have access to in the lab, in the doctor's office, in the field, wherever you're taking your measurements. And then now that we have Y and we have A, the measurement matrix, we can hand this off to this reconstruction algorithm that does L1 minimization, get a G hat, an approximation. We use FASTA, this FASTA algorithm to recover a G hat. And you can see the G hat that it gave as an approximation in the level order looks pretty darn close to the ground truth. This is the ground truth that we were trying to discover. We discovered the hidden gene network, and the goodness of fit is pretty good here compared to the ground truth. And the predicted full trait, we can now take the gene network, take the inverse Fourier transform, and predict the full trait for all genotypes. And this is what the predicted full trait T hat is compared to the ground truth. And you can see it looks pretty close. It's not perfect. We have a little bit of error. We could take more measurements, we get a better fit, fewer measurements. It won't be quite as good. And we can play some tricks. If we have a priori data, a priori information, we can maybe use a weighted L1 norm to give more weight or more bias to the lower levels. But essentially, we've achieved our goal of using compressive sensing to discover the gene network. And from that, we can do all the Fourier tricks that we talked about before. Here's the link to our paper. It's on the archive. I do want to point out that the new version won't be available until midnight GMT, which is in about 6 hours. So please don't click on this link for another 6 hours. There's an old version, but the new version of what I talked about today is a complete rework of the old version. So the new version will be available in about 6 hours. Thank you very much for your time.
All right, let's thank the speaker.